This video abstract is AI generated. Can a small molecule inhibit enzyme 1 without binding the active site? In this work, we study bacterial enzyme 1, the first protein in the phosphotransferase system, or PTS. Because enzyme 1 sits at the top of the sugar uptake pathway and has no close eukaryotic homologs, it is an attractive antimicrobial target. Our goal was to discover small molecules that inhibit enzyme 1 through an allosteric mechanism. To do that, we used an NMR-based fragment screening pipeline. We started from a library of 1000 rule of 3 compliant fragments. First, ligand-detected STD NMR identified fragment pools that bind enzyme 1. Next, protein-detected chemical shift perturbation experiments validated binding to the EIC domain. NMR titrations then measured binding affinities, and enzymatic assays tested whether the fragments inhibit phosphoryl transfer. This workflow narrowed the library to 6 low millimolar EIC ligands. Among them, 3 compounds inhibited enzyme 1 activity. The kinetic data showed that the 3 inhibitors do not behave identically. Compounds 1 and 2 act as competitive inhibitors. They increase the apparent K sub M for PEP binding. Compound 3 behaves as a mixed inhibitor. It increases K sub M and also decreases Vmax. Why do these compounds behave differently? NMR-CSB mapping and molecular docking revealed the answer. All 3 compounds bind the same previously unidentified surface pocket on EIC, located between alpha helix 1 and alpha helix 2, more than 10 angstroms away from the PEP binding site. Compounds 1 and 2 occupy this allosteric pocket and contact residue V292 near beta strand 2. These interactions perturb the PEP binding region and weaken PEP affinity, explaining their competitive behavior. Compound 3 also contacts V292, but it makes additional interactions with residues at the C-terminal end of alpha helix 2, especially S326 and Q327. Molecular dynamics and principal component analysis indicate that these extra contacts perturb the beta-3 alpha-3 active site loop and shift the enzyme toward less catalytically competent conformations. This explains why compound 3 shows mixed inhibition. Together, these results identify a remote allosteric pocket on enzyme 1 and show that binding at this site can inhibit catalysis through distinct mechanisms. This newly identified pocket provides a foundation for developing second-generation allosteric inhibitors of bacterial enzyme 1. To know more, read our work online.